Hi, everyone. Today, we're going to be going through Laboratory 2 for microbiology. We're going to start to develop an unknown bacterial or fungal culture, and we're going to be taking a look at some bacteria and fungi under the light microscope. So first of all, the way you're going to prepare some unknown samples is that you're going to be given two different types of agar plates. One plate will contain agar containing the nutrient agar. This is sort of a broad spectrum type of bacterial plate. It supports the growth of most bacterial types of species. And then we have Sauberod's agar. This one is preferential for fungi, and molds. So this one prefers fungi and molds, and pretty much is going to discourage the growth of many different types of bacteria. So as we've discussed in lecture, media provides a base support, especially this agar media, provides a base support for microbes to grow and provides moisture as well as nourishment. The type of nourishment provided will be selected for different types of organisms. As I said, nutrient agar is much more for bacteria, and Sauberod's agar is much more for fungi and molds. So you want to label the bottom or the agar side of the plates with your name, the date, and the lab section that you are in around the periphery. So, it should look a little bit like this, where you're going to put your name, date, lab. And you can also do some sort of ID if you need it. I think that most of the plates are labeled as whether they are nutrient agar or Sauberod's agar. So nutrient agar is labeled NA, and Sauberod's agar is labeled SA. So the nutrient agar plate is a plate that can actually grow very many different types of microorganisms. And because of that, it's suitable for many purposes. Most of the time when we're in this lab, we will be using nutrient agar plates. And if we're not using the solid surface, we'll be using the media, the nutrient media in a broth. So it would be nutrient broth. And that's the liquid form of it. So you can actually have the choice of whether you're going to treat the plate in a variety of different ways. You're going to touch the plate with your fingers, for example, and then close it, brush your hair over the plate, then close it, place coins on the plate, remove them, then close it, leave the plate open on your bench for 30 minutes to an hour, like for the duration of the lab, and then close it, leave the plate open in the hallway, somewhere where it can't be stepped on for 30 minutes to an hour, and then leave the plate with the top on and don't open it and see what happens. So these have, the agar has all been sterilized. But there is the possibility that when the plate is being poured, that sometimes a contaminant can get in, and over a period of time, it can grow. So those are different possibilities. You can also, think of some other way to treat the plate. Okay, so the Sauberod's agar plate, this is the second plate. It's labeled usually SA. This is one that has a little bit more dark color to it. It's a little bit more orangey or amber colored. This is a media that's used to preferentially grow fungi. It's not that it won't grow any bacteria, but generally fungus and molds prefer to grow on the Sauberod's agar. And the reason why is that it has a particularly low pH, which is really unsuited for bacterial growth. In addition, it contains gentamicin, which is an antibiotic, which is often used to treat gram-negative infections. So with this plate, you will prepare it by exposing the media to air in the laboratory for 30 to 60 minutes. So you're just going to leave this plate open. In the lab for 30 to 60 minutes. So you're going to have a variety of different types of slides to study. For example, you're going to be studying Klebsiella pneumonia. This one actually is on demonstration in the lab, just because it's really difficult to actually see this one. It can be used under the 45x objective, which would mean your final viewing magnification will be 450 times, or it can be used with the oil immersion lens at 100 times magnification, which means your final magnification will be at 1,000 times. As we've talked about before, the oil helps to capture and channel refracted light rays from the specimen. Please follow the directions of your instructor and the information at the end of the handout to learn proper technique for utilizing the oil immersion lens. We prefer that the oil immersion might not be used this particular lab, just because it is an extra level of complication and it makes the slides a mess. They're full of oil, and that means that it's difficult for people who come into the lab next, because they will have to use slides that have been affected by the actual use of the oil if they have not been properly cleaned. So with oil immersion, we place the slide on the mechanical stage. We focus on the point of focus on the lowest objective lens and center the point of interest. So, when you're on that 10x objective lens, you want to make sure that your sample is pretty much centered. And then that will enable you to, when you go to the next highest level, sorry, it's usually a 4x. I apologize there. This is usually 4x. So then when you go up to, the 10x objective, hopefully, you won't have to recenter your specimen very much. It will be pretty much centered. But if you didn't center it after the 4x, during the 4x, you're going to have sometimes a hard time as you go up in magnification finding your specimen. And then you should only just use, between each one of these, you should only use the fine focus. You should not use the coarse focus anymore. So, you can use coarse focus to get it into focus to the 4x, but then after that, just the fine focus. And so now you're going to go up to the 450. So hopefully, it's a 45x. And then what you will do is you'll move the actual 45x lens out of the way. And here would be your 100 times magnification. And here is your sample. And what you would do is actually, when you move it out of the way, then you would just put the oil on top of that. So the oil would go on here. And so then when you move the 100x into position, here's your specimen, the oil will actually make sort of a conduit, a pathway for the light to travel. And that way, it's going to capture all of the light rays. So then, when you go to the 100x, hopefully your sample is right in the middle, and you can see it more clearly. So it's really critical. Do not, so in each one of these, you only want to use fine focus. don't use the coarse because you could break the slide or break the lens. Also, you only want to use oil on the 100X. So only oil, on the 100X oil immersion lens. The other lenses are not designed for the purpose, and you will damage them. So that's very critical. So for more information on using the oil immersion lens, please visit that YouTube address. Thank you. So today we're going to be looking at the following. We're going to look at some fungus, which is yeast. And this is Saccharomyces cerevisiae. This one is a yeast that buds. This is also referred to as baker's yeast. So if you've ever made bread from scratch, you would have used baker's yeast to help the bread to rise. So it's used in bread baking as well as alcohol fermentation. As we've discussed, we said that Louis Pasteur adds it to grape juice in order to develop wine. So this is commonly used, and there are certain yeasts that actually have been developed specifically for making wine. So it's interesting because yeasts bud. So if you want to see how they reproduce, they actually produce a little bud off of the main cell. And so you can actually see this in some cases. You can see this cell is budding right there. And then, basically the smaller one comes off of the larger one and then grows. So you can see a lot of sort of what it looks like to be, different sizes of cells. And it's true, the younger cell, that's the newer cell, the younger cell, which is the bud. So another type of yeast is called Candida albicans. And this one is found in normal, healthy gastrointestinal tracts, as well as the female urogenital tract. Under certain circumstances, it actually could produce pathological conditions if it grows too much. Sometimes if it gets into the mouth, it actually causes a condition called thrush. And it's an infection of the mucous membranes of the throat and the mouth. There's also vulvovaginitis, which is basically when the yeast becomes too numerous in the vulva and the vagina. So this is an example of the yeast that you see here. And you can see it has a lot of similarities to the yeast, the actual baker's yeast. Another type of fungus that actually we're going to look at, and actually this one's a really pretty one. This is a Rhizopus nigricans, and it's a fungus that causes the fungus on bread. So this is the bread mold. And what you can see is this produces a whole bunch of spores, and these are not endospores, but these are just spores. And these spores are reproductive. So they produce these reproductive spores that produce more of the yeast. So it can not only be found on bread mold, but it can also be found on old food, in soil, and also very commonly in children's sandboxes. So we're going to be looking at a variety of different types of bacteria. One example is the Streptomyces. There are more than 500 different genera of this gram-positive bacteria. There are more than 500 different species. In the genus Streptomyces. They resemble fungi in their pattern of growth. Like fungi, they form these things called mycelium, sort of this very filamentous and branching. So that's sort of typical of fungus, but when they look at Streptomyces, even though it's a bacterium, it has a sort of a structure very similar to what you would find in fungus. So this one is a soil bacterium, and actually Streptomyces griseus produces an antibiotic called streptomycin, and we'll talk about that a little bit later in the lecture. So bacteria, this one is Klebsiella pneumonia, and this bacterium is part of the normal flora of the intestine in a small number of individuals. It is a gram-negative rod, and these organisms can produce a capsule that makes them particularly resistant and invisible to the host's immune system. So this one's only on demonstration only. The spaces around the bacteria, so you can sort of see these, this white space around the bacteria. So this is the bacteria. And you can see the white space. The white space is the capsule. So this area is the capsule. And the capsule, just like it is invisible to our immune system, it also does not stain very effectively. So it's very hydrophobic and it repels stain. Another type of bacterium that you'll be looking at is something called Diplococcus pneumonia. This one is a gram-positive bacterium that is characterized by these sort of coupled spheres. You normally see them together in twos. So that's why it's called Diplococcus, referred to as two spheres. And again, it is a causative agent of pneumonia. It is very frequently referred to as pneumococcus pneumonia or streptococcus pneumonia. This organism is often one of the causative agents of pneumococcal infections, including acute sinusitis, osteomyelitis, septic arthritis, and bacterial meningitis in adults, children, and dogs. So it does include pneumonia as well. Another bacterium, this one is Treponema pallidum. This is characterized as a gram-negative spirochete. It forms these spirals that you can see here. This is an organism that causes the sexually transmitted disease syphilis. It's characterized by a very fine and slender spiral. This one too is only under demonstration. The demo one, it doesn't show very clearly the spirals. It seems that when we prepare this bacteria, it sort of stretches out and looks more like what we see in this part of the slide. Okay, so when you look at it under demo, it does not have this nice, clear spiral, that is more typical of what they're supposed to look like. Another bacterium that you're going to be looking at is a particular type of bacillus, and there's many species in this genus. So this is a gram-positive rod, and it produces endospores. So remember, the endospores, they're not reproductive, but they are a dormant form of the bacterium. So these endospores are highly resilient. They're dormant forms of the organism that can often withstand many different adverse conditions, such as extreme desiccation for long periods, so that's drying out, and they still can germinate when living conditions are such to provide nourishment and good growing conditions for these organisms. Some of them will be anaerobic. But the endospores can withstand the presence of oxygen. Some are aerobic, and we will be using one in the lab this semester called Bacillus megaterium. The one that you're going to be looking at in the laboratory is called Bacillus subtilis. And that's pretty much what we're showing here. The pink part is the vegetative part of it. Those are the pink. It's the vegetative cell. While that sort of bluish green are the endospores. Some of the endospores have been released. Some of them are still contained within the cell. Another bacterium you're going to take a look at is Staphylococcus aureus. Staph aureus is a bacterium that is associated with the skin. The name directly translates to golden cluster seed. That term staphylo, I think we've talked about this in the lecture portion of the course, refers to sort of a grape-like structure. And that's because we often find these particular cells sort of hanging as if they were, grapes on a tree, or grapes on a vine, I should say. You sort of have this kind of congregation of the cells. So when you take a look at these cells, they cluster. So this is this grape-like clustering of cells. And that's why they're referred to as Staphylococcus, coccus, because they are spheres. So this can cause a range of illnesses from minor skin infections such as pimples, impetigo, boils that are also referred to as furuncles, cellulitis, folliculitis, carbuncles, scalded skin syndrome, and abscesses. They can cause very severe diseases, including pneumonia, meningitis, osteomyelitis, endocarditis, toxic shock syndrome, chest pain, bacteremia, and sepsis. So these are serious, dangerous types of bacteria. They should not be handled. And that's why we only have a prepared slide of them. We are not actually going to be working with the live bacteria. It is also the causative agent of the dreaded MRSA, which is methicillin-resistant Staphylococcus aureus, as well as VRSA, the vancomycin-resistant Staphylococcus aureus. Some of these strains of the organism have acquired genetic resistances to various antibiotics, such as the penicillin-like drug methicillin and the robust drug known as vancomycin. So in those cases, you have to use other types of antibiotics to help control those infections. All right, so that ends our discussion. I hope you enjoy looking at the slides. Thanks very much. Have a great day.